Hi, and welcome to today's Daily Chronicle. Today is Monday, March 2nd, 2026. Murder, Magic, and Medicine. John Mann. Murder. Toxic plant and animal products have been used for thousands of years in hunting, execution, and warfare. Usually, the poisonous extracts were smeared on arrows or spears, and the earliest reliable written evidence for such use appears in the Rigveda, an ancient poetic work from the Vedic period in India. The ancient Greek word toxicon originally meant poison for arrows. Both Homer and Virgil mention the use of arrow poisons derived from natural sources. For example, in the Odyssey, Athena reports that when she first met Odysseus, he was seeking the deadly poison, probably aconite, wherewith to anoint his bronze-tipped arrow. Aconite was the most widely used arrow in poison in medieval Europe, and was still in use on the Iberian Peninsula as late as the 17th century. Despite the potential for these toxins as agents of warfare and other murderous activities, they were originally intended as important hunting accessories. The arrow poisons of South America and Africa provide excellent examples of this. Arrow poisons. In the 16th century, explorers reported that the Indians of Brazil, Peru, Ecuador, and Colombia were using arrows tipped with wurari or wurali, local names for what we now call curare. This is a crude, dried extract of the plant Condo tomento, usually an admixture with various Skycnos species. Vivid accounts of the efficacy of these arrows were related by, among others, the Spanish explorer Francisco de Drugs that we take. Francisco de Orellano. The Indians killed another companion of ours, and in the truth, the arrow did not penetrate half a finger. But as it had poison on it, he gave up his soul to our Lord. And Sir Walter Raleigh, the party shot, endureth the most insufferable torment, and abideth a ugly and lamentable death. Here is the plant. The Macu Indian hunters of Colombia have a reputation for their potent curare preparations. But in the main, fantastic and largely misleading tales reached Europe. In particular, myths arose about the modes of preparation of these poisons and their efficacy. Was believed to be somehow related to the type of storage vessels used, in example, tube, pot, or gourd curaries. Between 1799 and 1804, the explorers Alexander von Humboldt and Aim von Platt surveyed the headwaters of the Amazon, navigated the rivers of Orinoco and Negro, trekked ancient Inca trails, and climbed several mountains. Von Platt also found time to report on more than 6,000 mostly unknown plants. Interestingly, despite the undoubted rigors of these expeditions and their exposure to tropical diseases, both of these expeditions and their exposure to tropical diseases, both of these intrepid adventurers lived to a ripe old age. '90 and '85 respectively. They also provided the first accurate account of curare preparation. It involved making an aqueous extract of the bark from the vines with a subsequent concentration in a yieldator-like mass. This was classified as a one-tree curare. If a wounded monkey could make only one leap before expiring, 3 tree curare could be used to capture animals alive. There was still total ignorance concerning the mode of action of the toxins until Charles Waterun began his experiments with the animals in the 1820s. His crucial experiment involved an ass which appeared to expire 10 minutes after he administered curare, but was then revived by artificial ventilation of its lungs using a bellows. The animal went on to make a full recovery, and the obvious implication was that the curare caused death by asphyxiation. In 1844, the French physiologist Claude Bernard, using nerve muscle preparations from frogs, confirmed that curare blocked transmission of nerve impulses to the muscles. Injection of the arrow poisoned into the bloodstream thus caused death by respiratory failure, asphyxiation, because the chest and abdominal muscles were paralyzed. He published these results and others in 1850. Much of the credit for the subsequent popularization and eventual clinical exploitation of curare must go to the American Richard Gill, who spent much of the 1920s living with the Indians in Ecuador and learning how to prepare and use the toxin. On his return to the USA in 1938, he carried with him around 30 pounds of curare. But his attempts to excite the interest of the various drug companies met with total failure, and in desperation, he set up his own curare business in California. An account of his adventures, White Water and Black Magic, was widely read, and eventually 2 drug companies, Squibb and Burroughs Wellcome, began experiments with curare. Early attempts to use curare as a muscle relaxant were carried out in conjunction with ether and anesthetic, and the experimental animals died of respiratory failure. In 1942, the Americans Griffith and Johnson first realized that artificial ventilation was necessary if the drug was to be used safely in conjunction with an anesthetic. In the same year, they went on to carry dozens of successful operations on humans using the new strategy. The most potent constituent of the curare, tubocurarine, was isolated by the Dr. Harold King in 1935. But owing to lack of fresh material, he was forced to use an old dry specimen of tube curare from the ceramics department of the British Museum as his source. This sample had been stored in a tube, and that was why the major constituent of curare was christened tubocurarine. The availability of the reasonable quantities of tubocurarine from the work of Squibb and Burroughs Wellcome allowed clinical evaluation to begin. And as was usual at the time, the human guinea pigs were company employees. Thus, Dr. Frederick Prescott, Wellcome's director of clinical research, was one of the first to have himself injected with a new drug. The sensational press reports, for example, Doctor died for 7 minutes, helped to ensure a speedy entry of the drug into clinical use. Supplies of curare still come from the South American jungles. Through isolation and purification of tubocurarine takes place in the USA and Britain. The Indians continue to use the crude plant product as highly effective arrow poison, even though firearms are now widely available. Interestingly, and fortunately for these native hunters, curare is poorly absorbed via the gastrointestinal tract, so consumption of the prey killed with the toxin is not hazardous. The mode of action of tubocurarine involves disruption of the normal functioning of one of the major neurotransmitters, acetylcholine, and it is now worth spending a few moments considering how this was achieved. A typical nerve cell, or neuron, is illustrated. It comprises a cell body of varying diameter, 5 to 100 micrometers, and a fiber, or axon, which usually branches extensively into dendrites. In humans, the axon varies in length from less than a millimeter to almost a meter, and in diameter from 1 to 25 micrometers. Although some of this thickness can include a non-continuous protein coat. This coat is termed to be the myelin sheath and functions as the electrical insulator. At rest, the neuron has different concentrations of ions. Groups of atoms that carry an electrical charge because they have lost or gained electrons on the inside of those in the surrounding fluid. In particular, there are many more sodium ions than potassium ions on the outside of the cell. And this situation is reversed on the inside of the cell. Excitation of a neuron is caused by local changes in the concentrations of various neurotransmitter substances and or certain ions, and as a result, there occurs a selective in a rush of sodium ions via special sodium channels. This leads to a change in the electrical potential difference of the neuron from negative 70 millivolts to around positive 40 millivolts, all within the space of a millisecond. At this point, the sodium channels close and separate potassium channels, open to allow an outflow of potassium ions and restoration of the original electrical potential difference. The nerve impulse is thus propagated as a series of depolarizations and repolarizations. In order to attain its original state, the neuron must now pump out the excess sodium ions and allow the uptake of potassium ions. And this must be accomplished before it can accept another impulse. There is no direct connection between nerve fibers or between nerve fibers and muscles or glands, and the junction between these cells, the electrical signal, is converted into a chemical one. The electrical signal causes the release of a neurotransmitter substance, which allows the gap between the two cells for interneuronal communications. This junction is known as a synapse, and other cell types it is termed as neuroeffector junction. The neurotransmitter binds to a specific receptor site, rather as a key fits into a lock, as depicted in the illustration. These receptors are usually glycoproteins. Of specific three-dimensional structure located on the surface of the postsynaptic cell membrane. Many drugs and other xenobiotics also fit into the various types of receptors. Those that elicit a response are termed agonists, and those that block the receptor site without initiating a response are called antagonists. The neurotransmitter thus attaches itself to a receptor on the next neuron and initiates a new electrical impulse, or binds to a muscle end plate or innervated gland and causes stimulation or inhibition. How these changes are affected is not completely understood, but alterations in cell membrane permeability to calcium, sodium, potassium, or chloride ions are probably involved. The neurotransmitter is then destroyed by the action of destructive enzymes or may be absorbed into the neural cell from which it was released. Many kinds of drug and other xenobiotics exert their effect by preventing or increasing the release of neurotransmitters. They do this by binding to the receptors, thus denying them access, by changing the permeability of the various cell membranes involved, by mimicking the action of a neurotransmitter, or by altering its reuptake by the neuron. Acetylcholine. This chemical is undoubtedly one of the most important neurotransmitters. It interacts with two subtypes of postsynaptic receptors, the so-called muscarinic and nicotinic receptors. The names reflect the fact that the natural plant substances muscarine from the fly agaric mushroom and nicotine from the tobacco plant will compete with acetylcholine for binding on their respective receptors and also cause stimulation. At certain neuronal junctions, notably in the gut, lungs, and heart, excitation elicits the release of noradrenaline, a structural relative of the most familiar adrenaline. Will stimulation of the adrenal glands, which lie just above the kidneys, produces adrenaline. These two neurotransmitters bind to so-called adrenoceptors, of which at least three main types have been identified, and elicits a plethora of responses. These responses are those due to acetylcholine, can best be explained pictorially as shown in the illustration. Nerves leaving the brain and innervating all organs, but not skeletal muscles. Which are under voluntary control, are part of the autonomic nervous system. These two types of nerves are identified primarily on the basis of their anatomical origins, though there are also pharmacological differences. Sympathetic and parasympathetic nerves are response to a threatening situation. The so-called fight-flight reaction provides a good example of the results of stimulation of the sympathetic nervous system. Adrenaline and noradrenaline are released and act upon the heart, lungs, gut, and peripheral blood vessels. As a result, the heart rate increases, the bronchioles. Numerous fine air passages in the lungs are dilated. From the gut to the skeletal muscle. In addition, or pupils dilate from the gut to the skeletal muscles. Our hair may stand on end, and constriction of the peripheral blood may lead to pallor. In contrast, when we are relaxed, stimulation of the parasympathetic system of acetylcholine is predominant. In contrast, when we are relaxed, stimulation of the parasympathetic system by acetylcholine is predominant. And the heart rate falls while blood flow is diverted to the gut to facilitate, for example, digestion. Clearly, our normal functioning relies to a great extent upon a delicate balance of these two parts of the autonomic system. Any xenobiotic that can upset this balance will produce a pharmacological response. With resultant changes in overall biochemistry. Our awareness of what is happening within us depends upon the magnitude of these changes. So returning to the mode of action of tubochorinine, the toxin binds to the motor end plate receptors and thus denies access to the neurotransmitter acetylcholine, with resultant paralysis of the muscles. When used in surgery, it allows a reduction to be made in the amount of general anesthetic required to achieve complete muscle relaxation, though the resultant paralysis of the respiratory muscles means that the artificial ventilation of the patient is necessary. Typically, a dose of 20 to 30 milligrams produces a paralysis lasting for around 30 minutes. A number of other toxins have been isolated from the various curare preparations used by South American Indians, and coxtophorine from Strychnos toxifera is probably the most important after tubochorin. It is about 25 times more potent than tubochorin and has a longer duration of actions when used in surgical practice. Though both drugs have been largely superseded by otherworldly synthetic neuromuscular blocking agents, such as pancorium and atracorium, these have a rigid molecular structure with two nitrogen atoms, which are positively charged under physiological conditions. The nitrogen atoms are held in a similar spatial arrangement to that found in tubochorin, and the distance between the two cationic centers is approximately the same. The drugs can therefore mimic the biological activity of tubochorin by binding the same acetylcholine receptor. The positively charged nitrogen atoms bind to negatively charged sites on the receptor. The main advantage of the newer synthetic agents is their shorter duration of activity. The induced paralysis is thus more easily regulated. In addition, they have much less effect than curare on the acetylcholine receptor in heart muscle. These are of the muscarinic class and differ from those of the nicotinic class in skeletal muscle. In Africa, the arrow poisons were usually based on extracts of various Strophothus species, and the great Victorian explorer David Livingstone was among the first to describe the native use of these plants. The major active principle from Strophothus and Strophothus combi, streptozidin, has an effect of the heart similar to the illicit of digitalis from the foxglove. A toxic dose produces nausea and vomiting, and marked a lowering of the heart rate and various heartbeat arrhythmias. David Livingstone also noted that a regulated plant species, Strophothus gratis, yielded a poison that could stop even large animals, like hippos and buffalo, in their tracks. The natives have developed various ingenious ways of piercing the tough animal hides in order to deliver the poison to the bloodstream. The active ingredient, ouabain, has similar effects to streptozidin, but at a much lower dose. Just as with the curare, and in contrast to digitalis, both arrow poisons are poorly absorbed via the gastrointestinal tract, so prey killed in this way is generally safe to eat. The mode of actions of these cardiac glycosides is different from that of curare. They all inhibit the functioning of a sodium-potassium ion pump that is needed for maintenance of the normal ion balance in the heart muscle cells. One consequence of this is that the concentration of sodium ions inside the cell increases, and this triggers a mobilization of calcium ions, which can, at therapeutic doses, result in more efficient muscular contraction. This effect is of considerable value in the treatment of congestive heart failure and of heart arrhythmias. In both states, the circulation of blood is impaired, and if not treated, the tissues, especially the brain, become starved of oxygen. Administration of cardiac glycosides increases the force of the efficiency of contraction of heart muscle, and so-called positive intraoptic effect. Cardiac output is thus made more efficient, and this is vital if the heart failure is to be reversed. In larger toxic doses, the cardiac glycosides cause serious disruptions in the ionic balance of the heart muscle cells, producing arrhythmias and ultimately cardiac arrest. The therapeutic benefits of cardiac glycosides from the foxglove were first described by William Witherings in the 18th century.